Meiosis I, the details. We just finished an overview of meiosis in the last video. The details are actually super important here because there are three things that make sexually reproducing organisms different from each other, or your kids different from you and different from each other. And that's super important for the process of evolution. You need to have a really variable population so that if the environment changes, the population, at least somebody from the population will live. So anyway, the details are super important because two of those things that make individuals different from each other happen right here in meiosis I. So the one that doesn't is two parents, and that's probably the one you knew before you took this course. But now let's talk about the other two. So here's what happens in meiosis I. You've got the nucleus, and the nuclear membrane breaks down. And so this is early prophase I. The stages of meiosis are the same as the stages of mitosis, sort of. You have PMAT, right? Prophase, metaphase, anaphase, telophase. The only difference is that with meiosis you have prophase I, metaphase I, anaphase I, telophase I. Then you have cytokinesis. Then you have prophase II, metaphase II, anaphase II, telophase II. So this is an overview of meiosis I, which is this stuff. So I bet you already know this stuff. Here they're at the middle, so that's metaphase. Here they're being pulled apart, so that's anaphase. And here's telophase at the end. So here's prophase, and at this point it's diploid and the chromosomes are all duplicated. The crazy important stuff happens here and here. So let's look at this next one. This is late prophase I. Let's actually just define them all first. Here's metaphase and anaphase and telophase. So what you've done here is separated out the homologous chromosomes. So one of the important things is that at the end of meiosis I, you've got haploid cells, but they're duplicated. So here the 2n number is 4. 1, 2, 3, 4. And here it's not 2n anymore. The n number is 2. You've only got 2 chromosomes, but they're still duplicated. So let's look at the details of what's happening in this. So let's look at prophase. So the thing that happens in prophase is something called tetrad formation or synapsis. And so what happens there is kind of actually unbelievably cool. The homologous chromosomes come near each other. And remember how if this is you making your eggs or your sperm, but this chromosome you had gotten from dad and this one you had gotten from mom. So your dad's chromosome, let's say right here, coded for blue eyes, and on your mom, that same area coded for brown eyes, let's say. Well, now look, this one is swapping a little piece with this one. And same thing here, they're swapping pieces. So there's early prophase, homologous chromosomes pair and they cross over. That's what you call it when they swap pieces, and I have, I think, a better picture coming up on that. In late prophase, the chromosomes condense, the spindle forms, the nuclear envelope fragments, all that stuff that you already knew about from mitosis happens. So let's look at the tetrads forming in prophase I. So you get homologous chromosomes and they come near each other. So here are the homologous chromosomes, and we're going to call that a tetrad. And so what you've got is this is a chromosome from dad and that's a chromosome from mom, let's say. And these are homologous chromosomes. So if this part right here codes for, your dad's trick knee, this one might code for your mom's normal knees. And if right here codes for, let's have it say a straight finger, and this one might code for a hitchhiker's finger or whatever. So these are homologous chromosomes, meaning they code for the same traits, but different variations of the traits. So here they are coming very close together, and that's called synapsis. And here's what it looks like. Here's the daddy chromosome, here's the mommy chromosome in you, right? This is your chromosomes, but this one had come from dad, that one had come from mom. They swap segments. So look at this way cool new chromosome. This sister chromatid is not exactly like that sister chromatid anymore, because it swapped that little spot. So this one is going to end up, it's a chromatid now, it's going to end up being a chromosome that's completely unique. It's a novel organization of DNA. And so you have DNA that was in your dad and DNA that was in your mom that now forms one chromosome. How cool is that? So homologous chromosomes in a tetrad cross over each other like that. And the pieces of chromosomes or genes are exchanged. And this actually happens before they condense up, but it's easier to show you like this. So this produces genetic recombination in the offspring. So you are different from your brothers and sisters and different from your parents. You have different, so you and your siblings have the same parents, but you got different combinations of that DNA. And let's see, you'll have the ability to form tons and tons and tons of different kids because your chromosomes will cross over and you'll give them different combinations of your parents' DNA. So here's just a picture of homologous chromosomes during crossover. And here's another picture of crossing over. So we're going to say this is maternal from your mother, this is from your father, and they're both in you. And here's what happens when you make eggs. You can sometimes have a double crossover. So this chromosome side, this sister chromatid that came from your mom, is totally like the one that's in your mom still. This one's different. This one has a little piece from dad, section from mom, and another section from dad. And this one's different, totally unique new combinations, but this one's just the same. So you can have a double crossover where two things cross, and you can have none crossover sometimes. So crossing over increases the number of different gamete types, which means the number of different variations possible in your offspring. Super, super important for evolution. So Metaphase I, they line up in the middle. This is the second important thing that you need to know about how organisms are different from each other. So homologous chromosome pairs line up along the equator of the cell, like this. And each chromosome in a homologous pair can line up on either side of the equator. We call that independent assortment. And I have a better picture of it coming up. So here we go. So you can see they've already crossed over. Here's the chromosome that was from dad, and here's the one that was from your mom. And they're lining up independently of these. See this one lined up with the mom's one here and the dad one's here. Look, it could have been the other way. You could have had the dad one here and the mom one here. So when these separate out, you're going to have different cells if they line up this way or if they line up this way. Your gametes will end up being different. And that's called independent assortment. So independent assortment meaning these assort, they separate out independent of those. So if the daddy one goes over here, this one, it could be either, it's independent, it could be either the mommy one or the daddy one from this one. So independent assortment increases the number of different gamete types, so the variations possible in your sperm or your eggs, which will eventually, may eventually become your offspring. And so in anaphase, they separate out and A for apart. And so at this point, notice they're still duplicated. You're separating out the homologous chromosomes, not the sister chromatids at this point. So this cell is going to end up getting a chromosome that was mainly from dad, and this one mainly from mom. But it could have been, these could have been swapped. It could have been the other way around. They line up independently. As long as the cell at the end ends up getting the right number of chromosomes, like one of chromosome 1, one of chromosome 2, one of chromosome 3, whatever it is. If if it's in you, one of each of your 23 pairs. As long as you end up getting one of each of those, they can line up however they line up. So homologous chromosomes separate and move to opposite poles. You can also say homologs separate. Sister chromatids remain attached at their kinetochores or centromeres. Telophase is just the end there where they separate out. So the nuclear envelope reassembles. Well, sometimes it does. The spindle disappears, and cytokinesis is going to divide these into two. So let's just review this. At the start of Meiosis II, you're going to end up with an N number, but they're duplicated. So if this were you, N would equal 23. This particular organism only started with 2 chromosomes, so now it has 1, but it's duplicated. It's 1N, and it's duplicated. If it were you, it would be N equals 23, but they would still be duplicated. So we're not quite done. We need the chromosomes to be not duplicated. So there's gene X. Only one homolog, or homologous chromosome, of each chromosome is present in the cell. Or I should say of each chromosome pair is present in the cell. Sister chromatids carry the same genes but different alleles if crossing over has occurred. So if there's no crossing over, this will be exactly like that. But if there's crossing over, then they might be different. Meiosis II, produces gametes with one copy of each chromosome and therefore one copy of each gene. So let's take a look at this. Here's the beginning of meiosis II. You've already separated out your homologous chromosomes. Now this is metaphase. The chromosomes line up in the middle. Anaphase, the sister chromatids have separated and now you have individual chromosomes. There's telophase and cytokinesis. And that's the end of the story. So meiosis II, you have prophase II, metaphase II, anaphase II, telophase II, and at the end you have cytokinesis. And you get 4 genetically different haploid cells. Haploid over here and duplicated. And at the end they are haploid. And the chromosomes, so the cells are haploid and the chromosomes are unduplicated. Prophase II, so here are the details of prophase II. Nuclear envelope fragments and the spindle forms. This looks a lot like mitosis, actually. Metaphase II, they line up in the middle at the equator, so the chromosomes align along the equator of the cell. Anaphase II, they separate apart. So it's the sister chromatids that are separating now. So sister chromatids separate and move to opposite poles, so you end up with unduplicated chromosomes at this point. And telophase II is when they separate out. So you get, from each one individual cell that you started with in the beginning of meiosis, you end up with 4 haploid chromosomes. So you make your nucleus again, your chromosomes decondense, they spread out, spindle disappears, and cytokinesis divides each cell into 2. And I think I'm going to pause.